Hi everyone and welcome to our model on lymphopoiesis 2. Lymphopoiesis is the process of formation of lymphocytes. We have two major forms of lymphocytes. This includes the B lymphocytes and the T lymphocytes. In this particular model, we'll be focusing only on the formation of the T lymphocytes. There's a separate model that talks about where we talk about the development of the B lymphocytes. As a form of overview for this particular model, in terms of the process of lymphopoiesis, it begins with the stem cells, what we call the hematopoietic pluripotent stem cells. The stem cells would eventually undergo cell division and differentiation to form committed cells or progenitor cells. And then finally, the progenitor cells will undergo further differentiation and maturation to form the precursor cells, which would eventually mature to form the mature lymphocyte, either B or T lymphocyte. In the case of lymphopoiesis, just to be more specific, the stem cells, pluripotent stem cells, will give rise to the T lymphocytic progenitor cells, and that would undergo further maturation to give rise to the T lymphocytes. And that maturation tends to occur within the thymus as compared to that of the B cells that occur within the bone marrow. So, when we're talking about each of the different stages of lymphopoiesis, particularly for the T cell, it's important to remember that this development tends to occur largely in the thymus and also out of the thymus, which is within the bloodstream and in the lymph nodes, as we saw in the development of the B cell. Starting with the stem cell stage, every form of lymphocyte will be derived from the pluripotent hematopoietic stem cell, just like every other blood cell within the circulation. The stem cell, the hematopoietic pluripotent stem cell, tends to differentiate into two major committed stem cells. This includes the myeloid lineage stem cell and the lymphoid lineage stem cell. Our focus in this model will be on the lymphoid lineage committed stem cell, which tends to eventually branch into the formation of progenitor cells for the T and the B cell lineage. Before we talk about the progenitor cell, it's important to note that the lymphoid committed stem cells has a very unique method of identification, and that is by the presence of a cluster of differentiation or receptor found on its membrane called CD10. CD10 is found in lymphoid committed stem cells that would either branch out to become the B cells or the T cells. So we don't really use the CD10 to distinguish between B and T cells. It is a marker found in all forms of lymphocyte or all forms of lymphoid committed stem cells. So we usually tend to call it the common all antigen. Um, Very important to note, lymphoid committed stem cells, because they are stem cells, they have the ability to undergo self-renewal and also cell division and differentiation. However, we cannot morphologically identify them. So the only way we tend to identify them is the presence of the CD10 on their cell membrane. And moving on to the progenitor cell, since we're focusing on the T cell lineage of lymphopoiesis, the progenitor cell under this particular lineage of lymphopoiesis is known as the pre-T cell. It is the first progenitor cell, and it is also found within the center of the hematopoietic inductive microenvironment, just like the pre-B cell. We use a different set of clusters of differentiation or receptors to identify the pre-T cell. If you remember, when we spoke about the pre-B cell, we mentioned clusters of differentiation 19, 20, 21, and 22 as the markers that we use in identifying the pre-B cell. For the pre-T cell, the clusters of differentiation or receptors we use includes the CD1, CD2, and CD3 receptors on the membrane. And very importantly to note, because the pre-T cell is a progenitor cell, it doesn't have the ability to self-renew. However, it is committed to the production of T cell. Well, it is not morphologically identifiable as a T cell yet. The pre-T cell tends to leave the bone marrow and enter into the thymus for further development. I will talk about the development next in the couple of slides to come. So at the thymus, the pre-T cell tends to exist in two major stages or forms. This depends on where exactly they are found within the thymus. The thymus tends to be divided into three major parts. We have the capsule of the thymus, then followed by the cortex of the thymus, shown here in dark colors, and then at the middle of this pale area is called the medulla of the thymus. So depending on whether they're found within the cortex or the medulla or at the capsular area, thymus, the T cells tend to be classified either as the pre-cortical T cell or a cortical T cell. So the cortical T cell are T cells found within the cortex, and then the pre-cortical T cells are the T cells found within the capsule. So the precortical T cells, as I said, are found at the pericortical area, just found outside of the cortex, very close to the capsule. They are T cells that express practically the same form of clusters of differentiation that is found in the pre-T cells found that are present within the bone marrow. So they have the clusters of differentiation 1, 2, and 3, and they have similar features as the T cells within the bone marrow. So they're just the similar to the T cells found in bone marrow, but the difference is that they are found at the pericortical area of the thymus and not at the bone marrow. So once a pre-T cell migrates from the capsular area of the thymus into the cortex, we begin to call them a cortical T cell. So cortical T cells are found within the cortex, and one very important distinction between the cortical T cells and the pre-cortical T cells is in the fact that the cortical T cells tend to lose its CD1 and gain CD4 and CD8. So when we are distinguishing them, remember that the cortical T cells has a CD2, CD3, and then a newly acquired CD4 and CD8 and lacks a CD1. So the precortical T cells, on the other hand, just has CD1, CD2, and CD3. Another very important thing that occurs once the pre-T cell enters into the cortex is a phenomenon we know as positive selection. So when I think about positive selection, I think about what occurs when we go through school. So imagine finishing primary school and then trying to transit from primary school to secondary school. We write a common entrance exam to enter into secondary school. And when we write a common entrance exam, people that pass the exam usually tend to be selected, and they're selected for secondary school, which is the next step. And whereas people that fail the exam would eventually need to stay back and rewrite the exam. So ideally, what happens in the cortex in terms of positive selection is that a T cell tends to be exposed to antigens within the cortex. These antigens are, um, are brought about or are presented by macrophages in the cortex. And the ability of a T cell to bind and interact with an antigen within the cortex would enable that T cell to be selected. So that's what we call positive selection. So T cells that are positively selected are T cells that have the ability to bind and interact with an antigen presented by macrophages in the cortex of the thymus. So once the selection occurs, these T cells automatically pass, and then they'll be selected to migrate from the cortex to the medulla, and they become what we call the medullary T cells. But T cells that do not pass positive selection, they end up being destroyed by the process of apoptosis within the cortex of the thymus. So moving on to the medullary T cells. Medullary T cells are the precursor cells of the T cell process or lineage of lymphopoiesis, what we call T cell ontogeny. Medullary T cells are T cells found within the medulla. They are T cells that are initially express CD2, CD3, CD4, CD8, and these are T cells just coming from the cortex. Remember that the cortical T cells have CD2, 3, 4, and 8. So when they undergo positive selection and enter into the medulla, they maintain these clusters of differentiation on their membrane. But then within the medulla, subsequently, the medullary T cells undergo two major important processes. The first is that they tend to lose either a CD4 or a CD8. So if they lose the CD4, they become a CD8 medullary T cell. So they have just a CD2, CD3, and CD8. Whereas if they lose CD4, if they lose the CD8 rather, they become the CD4 medullary T cell. So they would only have CD2, CD3, and CD4 clusters of differentiation on their membrane. And that's the first thing that happens to the medullary T cell. The second thing that happens to the medullary T cell is a process we know as negative selection. So positive selection occurs to cortical T cells within the cortex. Negative selection occurs to medullary T cells within the medulla. And what does negative selection mean? So negative selection is a process whereby the medullary T cells are exposed further to more antigens presented by macrophages in the medulla. Now, this is a bit different from what the positive selection. So, um, medullary T cells that are negatively selected are T cells that, although they're exposed and although they bind to the antigens, they are not going to undergo an excessive stimulation by that antigen. So once a medullary T cell is not excessively reactive to an antigen or avidly reactive to an antigen, then that T cell has passed the negative selection criteria. So think about it in a fashion of, we have two major T cells, two T cells, two medullary T cells. One of them is exposed to one antigen, the other was exposed to the same antigen. One of them binds avidly to that antigen and causes a reaction, starts to secrete um, different cytokines and eventually causes inflammation within the medulla. That T cell has failed the negative selection. The point of negative selection is to ensure that the cell is not overreactive. So the second T cell, on the other hand, binds to the receptor and um, exhibits an acceptable reaction, not too excessive, not too innate or inert rather. So as a result, this T cell is said to have passed the negative selection. So T cells that have the ability to bind to an antigen but do not avidly react to the antigen are said to undergo negative selection and would pass the process. So once a T cell passes negative selection, they move from the thymus into the bloodstream and then they become what we call the virgin T cells. So this is a process that explains for us the different forms of development of the T cell within the thymus. Starting with, we'll notice that T cells present within the thymus tends to be found first of all at the capsular area, and then eventually enters into the cortex. Within the cortex, they are exposed to different antigens presented by the macrophages. As you can see, the cortical epithelial cells or macrophages present different antigens to the T cell, and then these T cells, once they are presented with the antigens, will either bind actively or inactively to the antigens. Once they are able to bind to the actively to the antigens, they will say that they've undergone positive selection. So these T cells will further develop by entering into the medulla. Within the medulla, the T cells tend to lose their CD4 or CD8 clusters of differentiation. So we have CD4 specific medullary T cells as shown here, and then we have CD8 specific medullary T cells as shown here. Then these cells will further undergo what we call a negative selection. And once negative selection is done and the cell passes, the cells will eventually be taken into the bloodstream to become what we call the virgin T cells. So moving on to the virgin T cells, these are also called circulating T cells. T cells found within the bloodstream. They are cells that express CD2, CD3, and either a CD4 or CD8 cluster of differentiation. They possess a T receptor that has been tested and trusted by the thymus. And these cells, although they've been tested and trusted by the thymus, in the bloodstream themselves, they've not been antigenically stimulated. Hence the reason why we call them the virgin T cells. Once they are exposed to antigens within the bloodstream, they become antigenically stimulated and they are converted into a non-cleaved T cell. So cells that don't undergo antigenic stimulation remain as a virgin T cell within the bloodstream, whereas cells that undergo antigenic stimulation becomes non-cleaved cells and then they migrate to the lymph nodes, and we'll talk about that in the next slide. So the non-cleaved T cell are the T cells that eventually arises once antigenic stimulation occurs for the virgin T cell. These T cells are found within the parafollicular center of the lymph nodes. So just like the cleave cells of the B cell lineage, they express CD2, CD3, and either CD4 or CD5, just like the virgin B cells, and the virgin T cells rather, and they always tend to be found in two stages. The first stage usually is a small non-cleave T cell, and then that further matures to form is a large non-cleave T cell. Then eventually the non-cleave T cells would mature further to form the T immunoblast. T immunoblasts are the final precursor stage for the T cell ontogeny. They're found in the lymph nodes. They contain secretory T cell proteins that are important for the process of immunity. This includes the proteins like perforins, caspase, and calreticulin. They also express the similar closure of differentiation to the virgin T cells and the non-cleaved T cells, so CD2, CD3, and either the CD4 or CD8 cluster of differentiation on the receptor. Latent immunoblasts will further differentiate to become either of these following T cells, which are the mature cells. So they could either form the T helper cells, which is largely a CD4 cell, the cytotoxic T cell, which is largely a CD8 cell, and then other forms of subtypes of T cells. We have the T suppressor cells, the T delayed hypersensitivity cells, the T natural killer cells, and the memory T cells. Just a short note on the memory T cells. These are virgin-like T cells. They are T cells that appear like virgin T cells. The difference between them and the virgin T cells is that the memory T cells are cells that have already undergone antigenic stimulation, but just appear like the virgin T cells. They are cells that could automatically undergo a second set of antigenic stimulation, and then instead of just going through the process of becoming non-cleave T cells and T immunoblasts, they just mature directly into T immunoblasts, and then from there mature into their specific mature T cell. So at the end of T cell ontogeny, what we have is the formation of different forms of T cells, and these T cells have different functions within the bloodstream. So we have the medullary T cells eventually becoming cytotoxic T cells. Usually, cytotoxic T cells are CD8 cells that have the ability to mediate cell-mediated immunity and cytotoxic virus and tumor cell destruction. Then we have the T helper cells taking about 30 to 35% of the total amount of T cells formed through T cell ontogeny. The T helper cells are cells that tend to help the B cells in the production of antibodies, hence they are named T helper cells. Then we have T suppressor cells. T suppressor cells make up about less than 5% of the total population after lymphopoiesis occurs. They are cells that tend to limit immature hypersensitivity reaction, so hence the reason why we call them T suppressor cells. Then we have the T delayed hypersensitivity T cells. These are cells that make up less than 5% as well. They support and enhance macrophages functions against chronic bacterial and fungal reaction infections. And finally, we have the T natural killer cells. This also makes about less than 5%. They are identified, they tend to identify and destroy neoplastic cells, hence the reason why they are called natural killer cells. They target neoplastic cells, similar to what we see for cytotoxic T cells. So these are the different forms of T cells, and this is the outcome. The medullary T cells either give rise to 5 of these different forms of T cells, the largest being the cytotoxic T cells, followed by the T helper cells, and then the rest, which are minor subtypes of T cells. The entire process of the formation of T cells from the stem cell stage all the way down to the precursor and then the mature lymphocyte stage takes about 2 weeks or 14 days. Process of lymphopoiesis is regulated by a number of different factors. This includes the granulocyte colony stimulating growth factor, the stem cell factor, and interleukin 3. Let's conclude this particular module by talking about the different forms of pathologies that could occur in a T cell form of lymphopoiesis. We can have a problem with increased production of lymphocytes. This leads to a scenario we call lymphocytosis. We can have a decreased production of lymphocytes, which causes what we call lymphocytopenia. In case of malignancy of a pre-T cell and a medullary T cell, this leads to a form of leukemia we call an acute lymphocytic leukemia. And then in the case of malignancy of a virgin T cell found within the bloodstream, this could lead to a chronic lymphocytic leukemia. The difference is that the pre-T cell and medullary T cells are usually immature, so that's the reason why they present as acute lymphocytic, whereas the virgin T cells are mature, almost mature cells, and hence the reason why we tend to see them cause a chronic lymphocytic leukemia. And finally, malignancies of non-cleaved T cells, T immunoblasts that are found within the lymph nodes would lead to a lymphoma. And that concludes our module on lymphopoiesis 2.